The third speaker today, our last speaker, is Tamika John. Tamika got her undergraduate degree in biological sciences from the University of Maryland, Baltimore County, and then we were lucky enough to get her to come to Duke, where she's in the process of getting her PhD in molecular genetics and microbiology. Her mentor is Dr. Georgia Tamaris, and Tamika is going to talk to us about regulation of the CD8 T-cell response in HIV infection. Welcome, Tamika.
Thank you, Dr. Hicks, for that introduction. I'd also like to thank the organizers for inviting me to speak today. And before I get into my project looking at the effective response to HIV, I would like to first briefly review the need for HIV vaccine. The current methods used in treating those with HIV and preventing transmission, as many of us know in this room, they are very effective. However, they do present some difficulties, the greatest of which is adherence. In addition, women at the epicenter of the epidemic, the group most likely to be at risk for acquisition, many times do not feel empowered to regularly use or have regular access to some of these methods, including topical gels and condoms. So we really do need the HIV vaccine to combat issues that we have with adherence and access, as well as other issues with the current methods used. Previous research has shown that an effective vaccine needs to induce the neutralization and effective functions of antibodies, as well as T-cell effective functions of both CD4 and CD8 T-cells. CD8 T-cells will be the focus of the remainder of my talk. CD8 T-cell effective function has two arms. The first arm, which is depicted here, is when you have direct interaction of CD8 T-cells, which are shown in pink, and HIV-infected CD4 T-cells, which are depicted in blue. This direct interaction leads to the cytolysis or death of the HIV-infected CD4 T-cell. In addition, we also have a soluble arm in which a CD8 T-cell releases factors that act to inhibit virus replication in CD4 T-cells. The soluble arm will be the focus of the rest of my talk. We know from previous research that CD8 T-cells work to control the level of viremia in infected persons. This is crucial because the lower the viremia, the less likely an infected person is to transmit HIV. In addition, we know that the CD8 T-cell population is heterogeneous in its ability to inhibit virus replication. That is, that all CD8 T-cells are not created equally in their ability to inhibit HIV replication, and we must identify the right CD8 population, the specific features that control viremia. To achieve this end, our lab uses a population of virus controllers that were recruited from the Duke Infectious Disease Clinic. These persons are chronically infected, meaning that they've been infected with HIV for a period greater than 6 months, but they have low or undetectable levels of HIV within their system, although they are not now or have never been on therapy. So, pardon me, this group represents the gold standard of the CD8 effective response we want to elicit with the vaccine. However, the mechanisms at work for soluble inhibition of HIV are not well understood in this group. And that is actually the objective of the project that I'm going to describe. So the first question we wanted to address in order to characterize the CD8 response within this population is we wanted to determine which subset of CD8 T cells control HIV replication. Specifically, we wanted to identify the HIV antigen specificity of CD8 T cells that are able to inhibit virus via soluble mechanisms within this virus controller group. To do this, we take CD8 T cells isolated from virus controllers, and we stimulate them with different HIV peptide pools that are from different regions of HIV, including GAG, envelope, polymerase, and NEF. We then take these HIV peptide-stimulated cells and use it in our virus inhibition assay, which quantifies the CD8 inhibition of HIV replication. Or in other words, it tells us which antigen-specific subsets are responsible for the inhibitory activity that we see in these virus controllers. This graph represents the results of the virus inhibition assays I just described. Across the top, we have our different virus controllers, or VCs, and along the side, we have the peptide used for stimulation. A purple box will indicate antigen-specific CD8 T-cell inhibition of HIV. In example, if we look at our virus controller 30, we see that only when the CD8 T cells of this virus controller are stimulated with GAG2 do we have inhibition of HIV replication. Interestingly, when unstimulated, none of the CD8 T cells from any of our virus controllers are able to inhibit replication. On the other hand, if the CD8 T cells are stimulated with either the GAG2 pool, which corresponds to P24, which is viral capsid, or the NEF pool, we have most of the virus controllers, the CD8 T cells from virus controllers being able to inhibit replication. So we addressed the first question of which subset is responsible by identifying that GAG and NEF-specific CD8 T cells from our virus controllers are mostly responsible for the inhibition that we see. The next question is, what is the gene expression profile of these cells? In other words, what are these cells secreting to cause them to have this inhibitory activity? To address this question, I wanted to identify the genes differentially expressed in CD8 T cells with and without the ability to inhibit virus. This work was done in conjunction with, in collaboration with the Keen Lab. So to address this question, we took GAG2 stimulated cells because, again, when the CD8 cells from most of our VCs are stimulated with the GAG2 or P24 peptide, we have induced inhibition. We wanted to compare the gene expression of GAG2 stimulated cells and unstimulated cells because unstimulated cells from our virus controllers don't possess this activity to inhibit. We first had a focused look at, we first focused our look at the gene expression, and we looked at 3 cytokines, MIP1 alpha, MIP1 beta, and interferon gamma. So 2 of these, MIP1 alpha and MIP1 beta, are actually beta chemokines that our lab and others have shown to be involved in the CD8 response to HIV. Using other cohorts, they actually bind to one of the co-receptors to HIV, one of the co-receptors to R5 tropic HIV, blocking the entrance of R5 viruses, including transmitted founder viruses, to CD4 T cells. So when looking at the expression of these cytokines, we had 3 subject groups. The first group are HIV negative subjects. These serve to be a control because these subjects, whether their cells are stimulated or unstimulated, they do not exhibit any level of inhibition of virus replication. These subjects, control subjects, will be indicated by a red circle. The second group, indicated by a blue square, are the virus controllers that have GAG-induced inhibition. And the third group, indicated by a magenta triangle, are virus controllers without GAG-induced inhibition. On the y-axis, we have fold change in mRNA compared to unstimulated. That is to say, if we look at GAG2 stimulated cells, how much more mRNA do we find for the markers that are on the x-axis compared to unstimulated cells? The markers on the x-axis include our 3 cytokines of interest, as well as 2 controls, GABDH, a housekeeping gene, which we don't expect to see much change in the GAG2 stimulated versus unstimulated cells, and RANTES, a cytokine that was previously implicated in not playing a role in the CD8 response in other cohorts tested. So if we look at the fold change in mRNA across the board in our HIV negative subjects, we see that the average fold change is close to 1. This means that in GAG-stimulated and unstimulated cells, the amount of RNA for all of these markers is about the same, which is as expected. If we next look at our fold change in RNA for our VC groups with GAG-induced inhibition, we see for our controls, again, the value is near to 1, but when we look at our experimental cytokines, we see that there's a significant increase in the amount of RNA for these cytokines. If we look at our VCs without GAG-induced inhibition, we see that their profile is similar to that of our negative subjects. So we conclude that there's more RNA for MIP1 alpha, MIP1 beta, and interferon gamma within CD8 cells that are capable of inhibition. We next look to determine the mechanisms involved in regulating the antiviral response. That is, address the question of why is there an increase in RNA for these 3 cytokines that we observe. The first obvious response would be, well, there's an increase in transcription, which is a biological step that makes RNA. So we look to address this question. The layout of this graph is similar to last, in which we have our 3 groups, and we have our markers of interest along the x-axis. Along the y-axis, we have fold change in transcription rate compared to unstimulated. Again, this is GAG2 stimulated cells compared to unstimulated. With our negative control subject, we see again that the fold change is close to 1, saying that there's not a real change in transcription rate when these cells are stimulated with the GAG peptide. If we look at our VC with induced inhibition, we see that the fold change overall ranges from, some are close to 1, but most of them range from 2-fold all the way up to some being over 13-fold. This is saying that there is an increase in the transcription for our interferon, MIP1 alpha, and MIP1 beta within this population. Again, the VCs without GAG-induced inhibition has a profile that is similar to our seronegative subjects. So this tells us that there is an increased transcription rate that contributes to the increase that we see in RNA expression. Looking closely at the data, we're able to determine that the increased rate of transcription is not the only factor at play here, because there are some increases in the total amount of RNA that do not line up with the increased rate of transcription. So there are some other mechanisms at work. Our next idea was to look at RNA stability. When mRNA is made within the cell, the mRNA does not stick around permanently. mRNA is actually dynamic in that it is made and degraded at a very quick rate, especially for cytokines. mRNA degradation is tightly regulated because there are elements in the 3' UTR of many RNAs that can be bound by proteins that act to stabilize the mRNA, which means that it protects it from degradation. And there are other proteins that act to destabilize mRNA, which means that induce a rapid degradation of these mRNAs. It's known from previous studies that mRNA stability plays a role in the immune response. However, this is unexplored in the CD8 inhibition of HIV. So we decided to explore it. And this graph has a similar layout as the other two that I've shown you previously. The only difference is now the y-axis is fold change in RNA stability compared to unstimulated. With the seronegatives or HIV negative population, we see that the fold change again is close to 1 for all of the markers tested. For the VCs with induced GAG inhibitions, these are virus controllers with induced GAG inhibition, we see that the fold change, again, some of them are close to 1, but for many of them, they're over 2-fold change, which is significant. And for the VCs without GAG-induced inhibition, we see again their profile matches the HIV negative subjects. So we're able to conclude that there's increased RNA stability that contributes to the increase in RNA expression that we measured previously. So in summary, I was able to tell you today that in our virus controller population, NEG and GAG-specific CD8 T cells contribute to the control that we see within the population. We're building a current cytokine signature for these cells that includes interferon gamma, MIP1 alpha, MIP1 beta, and other cytokines that we've tested that I didn't necessarily have time to talk about today. And we now know that there's increased transcription and stabilization that leads to the increased expression of these mRNAs and others. I would like to thank members of my lab, especially Elise Frisbee, who's a technician that works closely with me on this project. And she actually has a poster number 14, that explores another arm of this project. I also like to thank the Duke CFAR because this project was actually inspired from another collaboration we had with the Keene Lab. During a meeting, we came up with this idea and we were able to explore this very interesting subject. The Ferrari Lab for providing reagents, the clinical core for aid within our patients, and as well as the Duke Adult Peds ID Clinic, the patients for participating, and the physicians who work to identify and recruit patients for us. Thank you.
Excellent. Do we have questions? Ken?
First of all, Tamika, congratulations. I think it's a good example of how a basic or translational scientist can interact with CFAR and through the adult ID clinic get specimens, and the specimens are really driving the research. So again, I think it's a shining example of the kind of things that we like to encourage. My question is, do you know if these cells are indeed polyfunctional, that there are individual cells producing multiple cytokines, chemokines? And if so, I think you've looked at CD107, a degranulation marker. How does that key out? Is that showing any significance at all?
So we were able, another part of this project, working with the Ferrari Lab, we were able to actually to run ICS and look at the polyfunctionality of these cells, and we do see that these cells are actually polyfunctional. I don't have the specific data because I haven't necessarily stain these cells for activation marker to say that the cells that are actually activated are those that are also polyfunctional. But we do believe that we are getting cells that are polyfunctional, and we're working now to develop more and more ICS experiments to look specifically at that, and to also isolate the cells that are the ones that are specifically activated so we can have more refined data.
Please, in the back.
So from your CD8 Tcells, the virus controllers, did all of them come from, or did all of them have the MHC class 1 haplotype? The ones that actually controlled the
Is the question, were all of them able to inhibit via MHC, or did they have the HLA alleles? No, they were not. Some of them did have beneficial HLA alleles previously identified as having benefit, but some did not.
May I ask a question, Tamika? I noticed on your graphs of the interferon gamma, MIP1 alpha, MIP1 beta responses, while the means and medians were clearly higher, there was a considerable range of values for individual subjects. Do you have a sense of why that's so, and how the ones with the highest degree of difference might differ from those who look almost like the controls?
Yes. So that's something that we're currently investigating. We think that it's different because these patients are different. They have different medical histories. They're from different backgrounds. What we're hoping to find is to find something that's common in those that are closer to the top that is not found in those that are closer to the bottom. So this is more of an initial step just to kind of parse out our groups so that exactly from population view, so we can then go in later and then look at again cells that are specifically activated, those that are not, and what are the signatures we see with those.
Great. Please.
To kind of dovetail the question, what was the range over time for your controllers? Were they controlling 2 years, 5 years, and onward? And would you be able to draw any the conclusions based upon the link of control?
So the range of control for our controllers range from some of them being recently affected, having been identified as a controller for 6 months, to those having been identified as a controller for many, many years. And again, this is the first exploratory step. That's another factor that we're considering as we try to parse out what's really going on in this system. Is it that those that have been controlled for many more years and have had a lot more exposure to antigen, are those the ones that we see more towards the top, having the high inhibition and having the high stabilization? It's a question that we're looking to answer, but we haven't reached that step yet.
Okay. Unfortunately, in the interest of time, we're about 10 minutes behind. I think I'll close off the questions. And will you be here, Tamika, so
Yes.
people can ask you questions later in the day? Okay. Thanks very much. 3 terrific presentations. I'd like to really think that was wonderful.